Good morning, ladies and gentlemen. I am Miss Prachi Vilas Kamble from Government College of Pharmacy, Karar. Today I am standing before you to speak some words on the topic Touch DNA, revolutionizing evidential DNA forensics. What comes to your mind when I say word Touch DNA? Touch DNA is defined as the skin cells which are remained on the object when we touch. Touch DNA is also called contact DNA, trace DNA, and the transfer DNA. Touch DNA technique used highly modified equipments which are very costly. That's why this technique is one of the costly techniques in the forensics. This technique is not even developed in our India. That's why Indians are not have even knowledge of this. This technique is useful in the cases like which have absent or in some small quantity of semen and blood, like murders or other cases. In these increasing cases of India, we have to develop this technique to identify the criminals time to time. This technique requires 7 to 8 cells which are outermost layer of the human skin. These cells containing DNA. This is collected from the equipments used by criminals or weapons used by criminals, their clothes and their personal items, and also from surfaces like table, handles of doors, and also cars. These are collected by the methods like swabbing methods. Swabbing methods are of three types: wet swabbing, dry swabbing, and double swabbing. In wet swabbing, a swab is collected and dipped into the water and then rubbed over the surface, that surface containing the DNA sample, and then collected it over. Second is dry method. In dry swabbing method, a swab is collected and then after the wet swabbing, small amount of sample is remained on the surface. This dry swab is used to collect the remaining DNA sample. In double swabbing method, both the dry and wet swabs are used to collect the DNA sample. Second method is tape method. In tape method, sticky tape is used to collect the DNA sample. Other methods like cutting and vacuuming method is also used to collect the DNA sample. These samples, after collection, then they are isolated, sterilized, and after this, various copies of DNA samples are made by the method used polymerase chain reaction, which is also called a PCR. After the copies made, these copies are used to identification of murderer or criminal. This method is useful to identification of who touched the objects at the crime scene, or to identify the criminal, or to solve the old cases, and also for reconstruct the crime scene. Also, the touch DNA. In touch DNA techniques, we have to face also various problems like small amount of DNA samples, various contaminations, and also the removal of various important samples. This touch DNA technology is used in USA in 2015, which is used in murder case. There is Alison Feldman, which is murdered, and at that case, no any obvious suspect is there. So there is Feldman's body and various other objects. We collect the data or touch DNA samples from that objects, and then we transfer it to the lab, and that causes the identification of murderer, not initially but after some time collection of DNA database, and the murderer was identified. In recently cases in India, murder and rape cases are increases, and semen sample or the blood samples are very rare to identify. So that's why this technique development in India is very important. Thank you.